Hello, everyone, and welcome to today's webinar, Design and Evaluation of a Multiplexed Assay to Assess Human Immunogenicity against Humira. This webinar is part of the Cell and Gene Therapy Virtual Event Series 2024, brought to you by LabRoots and is sponsored by Luminex. For more information on our sponsor, click the Luminex logo found on your screen. I am Israel Gonzalez of LabRoots, and I'll be your moderator for this webinar today. Before we begin, I would like to remind everyone that this event is interactive, and we encourage you to participate by both communicating with other attendees using the group chat and also by submitting questions during the presentation. Now let's get started. I'd like to introduce our speaker, Paul Rhyne, bioassay lead at the Bill and Melinda Gates Medical Research Institute. You can view Paul's complete biography below. Paul, you may now begin your presentation.
Hello, everyone. This is Paul Ryan from the Bill and Melinda Gates Medical Research Institute. And I just wanted to thank you for this opportunity to bring this set of work to you. And today I'm going to be talking about some work that we published a while back. And it has to do with multiplexing solutions for looking at immunogenicity. And we use Humira as the example for that. Let me just kind of walk you through some of these different concepts here. So if you remember your basic immunology, anytime you have a new antigen that is foreign that comes into your body, you basically get two different types of immune responses. You get these cellular responses and you get these antibody responses. And it's these antibody type responses that we're going to be focusing on in today's talk. So as far as the pharmaceutical industry, there are a class of drugs that are referred to as biologics. And this is sort of the newer type. And they can be any one of different types of molecules. They can be proteins, they can be tissues, vaccines, gene therapy products, allergen extracts, monoclonal antibodies. All of these are considered to be biologics. So the issue is when you use these biologics and put them into human beings, often your immune response will see these biologics as foreign, and they will generate an antibody mediated response against that drug. So on this cartoon we have here, we took the biologic Humira, which is a fully human antibody, and what you can see here is that once you give that to a person, if you get a humoral immune response, you get antibodies that bind to this drug, and they form these complexes, which are referred to as anti-drug antibody complexes. And that's really going to be the focus of the talk, is when this happens. So generally speaking, when you have antibody responses against these drugs, there are a lot of things that can happen. One of them is you can affect the drug efficacy. So these antibodies can bind to the drug and prevent it from working. And there are even alternatively, there are cases where these antibodies actually increase the drug's effectiveness. But often or not, they would prevent the drug from working, which is an unwanted side effect. The other thing that these anti-drug antibodies can do is they can alter the pharmacokinetic or PK profile of that. And I've got a quick figure here that just shows what that looks like. So this was published back in January 2018. What you're looking at here is the concentration of a biologic in the form of a monoclonal antibody was given to animals. And you can see over time that the levels of that drug slowly degrade as expected. In animals, however, that had an anti-drug antibody response, that's this orange dotted line here, what you see is that PK profile after about 20 days, the drug concentration drops significantly. And if you look at the presence of these anti-drug antibodies in these animals, you can see them basically where there were no anti-drug antibodies. And now by the time you get to day 20, day 30, day 40, you can see a sharp increase in these. So what's happening here is these antibodies are basically clearing the drug out of the animal system and altering PK. And this can happen in humans, and it's somewhat concerning if you get altered drug levels. So another consequence of this immunogenicity is what we refer to as these unwanted clinical effects. Some examples are those where you can get hypersensitivity type responses, you can get deficiency syndromes, and other things that can have severe clinical consequences. So one of those classic effects of this is when you have kidney patients that have anemia. So they have low levels of these red blood cells. And there is a miracle drug that was one of the first biologics. It's erythropoietin. So this is the purified protein that you can treat these patients. It goes into the bone marrow and basically stimulates red blood cell production and alleviates the anemia. So this was a life-saving therapy for patients that have this. Unfortunately, after some time, it was realized that sometimes these patients make an antibody response to that erythropoietin. And what that does is these antibodies will bind the erythropoietin that's being given to them and prevent it from working. But they were also cross reacting with the native erythropoietin that was naturally created. So instead of helping the patient, you basically have a complete crash. These patients went from having low red blood cells to almost no red blood cells, and that was referred to as pure red blood cell aplasia. And this is a life-threatening event. So it was realized early on by the regulatory agencies that these anti-drug antibodies can have severe consequences. So it's definitely something that we have to be measuring. So the way that this works, if you want to evaluate immunogenicity in your clinical population, you have to screen all of your patients and all of the samples for the presence of these antibodies that might be reactive to the drug. And this is referred to as a screening assay. And these assays need to be simple in concept and they need to be qualitative. So they need to be able to detect the presence of these anti-drug antibodies. And the way these screening assays work is you use something called a cut point. So you apply this to your assay, and this cut point allows you to distinguish between background noise in your assay versus a sample that really does have an anti-drug antibody in it. And this cut point needs to be set statistically so that there is at least a 5% false positive rate. The worst thing that can happen is that if you have a patient that has these anti-drug antibodies and your assay scores them as negative, that would be a false negative. That is not what you want. So we always move these statistically. So if anything, it gives a false positive rate. So how do these screening assays work and how do you generate this screening cut point? Well, the exercise is you take a large panel of clinical samples from drug naive patients, usually somewhere between 50 and 100. And you take these samples and you put them in your screening assay and you run them a bunch of times, at least 6 times. And you look at the values that these samples give you in your screening assay. And I have that plotted here in this cartoon. And that allows you statistically to set what's called that particular cut point so that 95% of these samples from drug naive humans will fall below that cut point and only 5% fall above it. And that's how these cut points are set. So when you go to analyze your clinical population for these anti-drug antibodies, you have this screening assay and you screen all your samples once. Samples that are negative or below that particular cut point are scored negative and the effort is over. Those that give you a positive signal, those that are above that particular cut point, you have to put them in another assay to basically confirm that there really are anti-drug antibodies in that sample. And you would do that with your screening assay, but you just add in purified drug to block that anti-drug antibody from generating a signal, and then you can confirm that it is a true positive. Samples that actually have these anti-drug antibodies in them that have been confirmed positive, you then have to go on and do further analysis of those samples. You usually have to titer those antibody responses so you can get an idea of the magnitude of anti-drug antibodies that are present. And sometimes you'll have to do an assessment to see if these antibodies are interfering with your drug's function. These are referred to as drug neutralizing assays. So you start with a screening assay and you confirm it, then you typically do a titer, and sometimes you have to do these drug neutralizing assays. And what we really need to do at this point in the talk is to try to describe you how these screening assays are done. And the first step is to take your sample and you're going to mix it with drug that is labeled with either biotin or sulfatag for the MSD technology system. And it's this mixture of these what's called drug conjugates that are placed with your sample. If there are antibodies that bind to your drug, there's some population of those antibodies that will capture a biotin labeled drug and a sulfatag labeled drug. And all of the biotin-labeled drug is going to bind to your plate surface, which is streptavidin. And then you give your sample a wash, and what you're left with are these trapped drug conjugate complexes that are bridged by these anti-drug antibodies. So now that that unbound material has been washed away, you add your buffers that are needed for this MSD technology, and you apply an electric current across that well, and it generates a signal via electrochemoluminescence. And it allows you to give a signal in your assay. Now, once you have a cut point defined on here, you can determine if this signal is above that cut point, and they would be screened positive or they would be screened negative. So this is a commonly used format on the MSD platform. But part of this talk is to show you that you can do this on other technologies. And in this case, this would be applied to the Luminex technology. So you can set up a screening assay on these beads in a very similar format. And the way we went about doing that is we took Luminex beads and we coated them with the drug itself, which was unlabeled. You mix these bead sets with your sample. If there are anti-drug antibodies in that sample, they're going to bind to these beads via that drug. So we know that there are drug-specific antibodies bound to these beads. You then come back with biotin-labeled drug, and you form that same bridge that we look for in the other screening assay format. And then you give it a wash, and then you finally come back with streptavidin phycoerythrin to give a signal. It's important to make sure that the drug is part of this detection process so that you can differentiate between a random human immunoglobulin from your sample versus a true anti-drug antibody. So that's why the drug has to be part of this. And this screening assay actually works very well, and it's just as sensitive as the other formats that are commonly used. So part of this immunogenicity profiling, you have your screening, you have your confirmation, confirmed positives, and you have your titer assays. Sometimes, and when you interact with these regulatory agencies, they're going to want to know what type of isotypes of antibodies are driving this anti-drug response. The guidance documents and the industry as a whole really doesn't give a lot of help with this. It just says that you need to isotype them, and it kind of leaves you on your own as to figure out how exactly that's going to be done. And that's the second part of this talk. And then we're going to combine everything into one multiplex assay. So isotyping, just to remind you of what we're talking about. If you go back to your basic human immunology and you look at immunoglobulins or antibodies inside of humans, you remember that there are different classes. So there's an IgM, a D, a G, there's an A and an E, and there are different variants or subclasses for each one of these. So for instance, the IgGs, there are 4 of them. So you have an IgG 1, 2, 3, and 4. There are 2 of the IgAs. So there's a whole mixture of different types of antibodies that are floating around inside of all of us. And these differ in concentration, they differ in size, and they differ in half-life. So these are often correlated with different types of clinical responses. And this is what sometimes the regulatory agencies want to know is there a specific isotype like an IgE that may be driving a hyperimmune response to your particular drug? So this is why you sometimes have to go about doing that. But now that we remember what these isotypes are, how do you measure an isotype in these screening and anti-drug antibody assays? Well, one of the ways that you can do this is you have to purify the antibodies out of your sample first, and then you can begin your isotyping. So how do you purify them? Well, you can take your sample and you mix it with biotinylated drug, and you allow the complexes to be formed, and you put them on a streptavidin plate. And that's basically going to capture your anti-drug antibodies. You give these plates a wash to remove the unbound material. And then what you end up with are captured or purified anti-drug antibodies on your surface of your plate. And if you add a low pH buffer, these antibodies can be popped off and they'll leave the drug biotin bound to the plate, but the antibodies basically become free in the solution, and therefore you have your purified anti-drug antibodies. And that's the approach that most of us take when we go to purify that. But once we have purified antibodies, now we need to isotype them. And the way that we did that was we took these purified antibodies and we mixed them with a Luminex bead that had an anti-Fc antibody already conjugated to it. And these anti-Fcs were specific for any given isotype. In this case, it would be an IgG1. So all of your IgG1 antibodies would be captured by this bead. And then you come back again with drug because you want to make sure it's drug specific. And you add your biotinylated drug to that, which will bind to these bound antibodies. And then you come back with streptavidin to give you your signal. So with this type of scenario, and you have your purified anti-drug antibodies, any kind of signal on this bead we know would be an IgG, or in the case for the previous slide, it would be an IgG1. So we have essentially isotyped this. But we don't have to stop with just one bead because we can leverage that multiplexing format. So in this case, we took Luminex beads that were already coupled with all of the different isotype antibodies. So there's a different bead set for each with each individual isotype and subclass. And these were multiplexed together. So when you take these purified anti-drug antibodies, you put them in this multiplex, we can tell exactly what isotype there are and if there's a mixture of more than one. So we can determine that. But let's get back to the whole flow of this talk. This was about multiplexing immunogenicity analysis. So you have to understand what's going on in the industry right now. They take these clinical samples and they perform these screening assays. And if they're scored positive, they have to go back to that sample, thaw it out, and run it again in the confirmation assay. If it's positive with that, they got to go back to the sample, thaw it out, put it in their titering assay. If they have to put it in a neutralizing assay, they got to go back to the sample, thaw it again, and put it in the drug neutralization assay. If they're required to do an isotyping, then they have to go back again to that same sample, thaw it out again, and put it in the different isotyping assays. If there's more than one assay, if you're looking at one specific type, or you're using an isotyping kit. So the problem that the industry is having right now is that this tiered approach is great, except that it plays havoc on your samples. So If we know that we can run these assays on the Luminex platform as well as the MSD and the colorimetric ELISA, why can't we multiplex them or combine multiple parts of these assays into one? It saves on time, it saves on sample, and you get more information in a more rapid response. And that's what the rest of this presentation just focuses on. So we decided to combine the screening assay and also isotype the any detected anti-drug antibodies in our screening assay. We can tell you exactly what they were. So we just put these two together in an effort to cut down on this. And the way that we did this was we took drug-coated beads, which I've talked to you about previously in the talk, and you have your isotyping bead. So you have 7 different bead sets, one for each of the different major isotypes, and you mix them together. And then you don't mix them with your sample. You mix them with purified anti-drug antibodies from your sample, and you allow them to bind to these beads. So if there are anti-drug antibodies present, they're going to bind to the Humira bead. And then you're going to be able to tell what isotype they are by which bead set they bind to for the isotyping portion. You come back with biotinylated drug. That's going to add a mechanism for detection. And then you add your streptavidin phycoerythrin to give you your fluorescent signal. So basically, we can now screen samples to see if there are anti-drug antibodies there, and we can tell you what isotype they are all in one assay without thawing the sample multiple times. So we did this. We did a lot of experiments. We set our cut points, and I've got a little bit of data of what that looked like in this particular slide. So with our screening assay, it works on a ratio of negative serum versus a known positive, and it's those ratios that drive these different cut point values here. And this is a histogram of the Humira bead on the lower left. These are all of the individual drug naive samples that we ran, and you can see where they're falling as far as on our assay. And you can see this blue line here is our derived cut point, which represents approximately 5% of these samples would be above that line and 95% would be below it. We did it for all of the bead sets, all of the isotyping bead sets. I just showed you one here for the IgG1. You can see what that cut point looks like visually, so that anything at or above this cut point would represent a positive value. So given that, we took all of our multiplexing approaches and our screening and isotyping assays, and we started applying that to different disease state samples. So this figure here shows 10 different serum samples that were taken from psoriasis patients. Some of them had been treated with Humira and others had not. And the way that you read this is you look across the top of this graph and you can see the different bead sets corresponding to what you're measuring, whether it's IgG1, IgG2, or anti-Humira. And then you have your derived cut point from our cut point screening analysis exercise. And what you're seeing here, if you just look at the Humira cut point 1.31, you can see that none of them were positive, at least at this point, for anti-drug antibodies to Humira. But we did get some positives from these different psoriasis patients that were above these different cut points. And then when we went back and looked at just the high values versus those that were at these different cut points, we still see that there actually were some IgGs that were coming across to that Humira that were being carried over. So it's not a perfect assay, and we were getting some positives. And when we went back and did confirmation assays on these were all turned out to be negative. So it does work in the end, but we did get some high signals, which is kind of what we would expect, although it should be 5%, but we got a higher percentage than that. But this is a small subset. So at the end of this did work for these psoriasis patients. So none of these were true positives, but we still had to deal with that 5% false positive rate. And as you can see, those that were really high turned out to not be confirmed. So in these psoriasis patients that were not, we didn't really find any anti-Humira antibodies in this particular set. Okay, so let's look at a different set. Let's look at rheumatoid arthritis patients. And in this population, we knew that some of these did and were treated with Humira. And of these 10 samples that we looked at, we had a bunch that were higher above the cut point for anti-Humira than others. And then we went back and confirmed those. So we only had two confirmed positives in these Humira treated rheumatoid arthritis patients. And of those that confirmed positive, we were able to find an IgG3 type response in one of them, and then a mixed response of IgG1, 2, and 3 in a different patient that had a high amount of anti-Humira. And again, these turned out to be a false positive for this, even though in this first sample it was 7.3 when it was way above that, but that turned out to be some sort of cross-reactive artifact that did not confirm. So all in all, what we can basically show you here is that you can combine a screening assay and isotype these different responses all at the same time. And if you go to the manuscript, it's got a lot more detail, a lot more data on how we did this, and a lot more explanation of all of these different samples. So just to kind of bring things back, you have to remember that biologics often generate unwanted immune responses when we put them into human beings. And these unwanted immune responses can alter drug function, they can alter the PK profile, and sometimes they lead to unwanted clinical side effects, which can be life threatening. So the regulatory authorities for these often require an anti-drug antibody assessment for all of these biologics, and that is that tiered approach where you have to do a screen, a confirm, and a titer type assays. And all of these work off different cut points because they're all qualitative results. Occasionally, sometimes, if there's certain clinical responses that are being noted, an isotyping has to be done in addition to that. And the other thing that we talked about was there was an industry standard for using these plate-based assays. But I just showed you that they don't have to be plate-based. They can be done on something like a Luminex platform. And what you can see here is that you can measure immune responses to biologics using these bead-based assays. And these immunogenicity assays, they can be multiplexed together. For example, in this particular presentation, we multiplex the screening and isotyping assay. But there's no reason why you couldn't combine the other assays at the same time. And then with the screening and isotyping assay, we found that the assay that we developed had excellent sensitivity and reproducibility, and that we found good results in both psoriasis and rheumatoid arthritis data, and it showed good results. So we had some false positives in there, and we were able to distinguish those from true positives. And with those true positives, we were able to tell you what kind of isotype response was being done there. So with that, I'd be happy to take questions, and I appreciate all of the time. And thank you very much.
All right, Paul, thank you so much for that informative presentation. Let's go ahead and dive into the Q&A section now. And I'm going to go ahead and encourage our audience to continue to submit your questions for our speaker to answer during this time. We do have several questions coming in, which is great. Thank you all for being active in the Q&A and the chat. So let's start with this first question, Paul. How much sample was used in both the Luminex and MSD platforms?
Yeah, that's a great question. We didn't do this study on the MSD platforms, but typically in MRD for an immunogenicity screening is somewhere around 1 to 10, maybe 1 to 20. You don't want to go too high, or you what you do is you risk diluting out your positive antibodies. So the regulatory agencies don't like you to go very high. So if you were to use an MRD of like a 1 to 4, you're really only looking at maybe 10, 15 microliters of your sample per well. So it's not very much. And with the Luminex assay, it's about the same. And the advantage is that with that small amount, you can get all of the isotyping done as well as the screening assays with that small amount of sample. So that is a definite plus.
Great. Thank you so much for that answer. Our next question is, in your psoriasis studies, have you used the Luminex assay to screen patient samples for cytokines to coincide with the isotypes?
Yeah, that's an interesting question. So we didn't apply this to psoriasis patients. We just developed this assay as proof of concept, and we chose the psoriasis um, patients because we knew that there is a, a lot of those are treated with Humira, and we felt like that would be the right disease state. Certainly, cytokines would be needed for that because you want to make one of the biggest issues is inflammation in your psoriasis, and you want to be able to correlate that with both the need to be treated by Humira and when you measure these anti-drug antibodies, it would be interesting to see what the different cytokine and the immune response patterns are during treatment and without, and if that correlates to any sort of increased levels of anti-drug antibodies. I don't know that that's been done too much, but that is a very interesting question. But we did not do that for this study.
Understood. This next question is kind of a two-parter. What were your anticoagulants used in the serum samples? And did you find any challenges in sample stability that might affect the results in these multiplex assays?
Yeah, so we'll deal with the first one. So these were serum samples, and these were mostly commercially bought samples. So serum doesn't typically have a coagulant inhibitor like a plasma sample would have EDTA or something like that. So these would be serum samples. And just repeat that second part of that question, if you don't mind, Israel.
Of course. Did you
Mhm.
find any challenges in the sample stability that might affect the results in these multiplex assays?
Yeah, so sample stability for antibodies is usually good for a couple of years, but that is so we did not find any stability issues with these, but we didn't take this out beyond 6 months after they were bought commercially. But you will run into stability when you have multiple freeze thaws. So if you subject these samples to repeated freeze thaw cycles for your screening, your confirmation, your titer, neutralization, and then you want to go on to do multiple isotypes. What you want to do is to use different aliquots of that sample so you can minimize that. But even with that, antibodies are usually pretty hardy. So stability is still something that you have to show, but I would say it would probably extend well past 1 year on average.
Great, thank you so much. Our next question is, a common challenge with ADA assays is drug tolerance and/or soluble target interference.
Yeah.
Could you share anywhere whether Luminex technology has successfully addressed these issues, and also what modifications or approaches help mitigate these challenges?
Yeah, so drug tolerance, for those of you not familiar with that, that is when in your patient samples you still have presence of the drug where these patients are being treated with, and the presence of that drug can interfere with the detection of these antibodies. So the drug tolerance for these assays was around 10 micrograms per ml. Actually, I think it was 50. So it was fairly high for Humira. One of the ways that the industry gets around that is that you do an acidification of your sample to help break up those complexes first, and then you add your drug conjugates to that acidified mixture. And then when you neutralize the pH, your drug conjugates go back into formation with those anti-drug antibodies. We essentially did that for this strategy because we had to purify the antibodies using an acidification step first, where we would acidify the sample and then we put it on a mixture with biotinylated Humira, which was captured on those streptavidin plates, and that would have gotten rid of most of your drug if there was any on there. It really doesn't matter the platform, MSD versus Luminex, as far as drug tolerance is concerned, particularly if you have these acidification steps. But certainly that is something that you have to characterize, you have to measure, and that is something that regulatory authorities are going to be asking for if you use these assays, whether it's on MSD or Luminex. So that's a very important thing that you have to address. It's hard to give answers to these questions when I'm not speaking to these individuals live, so I'm doing my best. So forgive me if I don't fully answer.
Yeah, and to the audience, if there is a follow-up question that you'd like to ask, if for any reason you need further clarification, you're more than welcome to do so by submitting the same question within the same question box there on the screen. But for now, let's go ahead and move on to our next question, which, by the way, Paul, we haven't gotten any follow-up questions, so I think you're doing pretty well.
All right, no problem.
So our next question is, what inspired the shift toward multiplex assays for immunogenicity testing, and what were the main challenges in developing this approach?
So there's two aspects to that. When you use an ELISA approach or an MSD approach, you typically do these assays one at a time. So you do your screening assay first. All of those are positives. You go on and you go back to that same sample, you confirm it's positive, and you keep going back and you keep repeating the analysis in a sequential manner. And that takes a lot of time for your lab to get through. So we figured that if you could multiplex some of these steps or combine them into one assay, that's going to save time and sample and make your sample analysis more efficient. And we've talked about this, and there's other similar assays that will use what we did in this Luminex approach. So we knew that this would work. So we put this paper together as an example of how to multiplex this. And we chose the screening assay with the isotyping assay, mainly because the isotyping assays, there is no guidance in the regulatory guidance documents that really tells you how to go about doing an isotyping assay. And multiplexing is really set up nicely for that. So that's why we combine those two. As far as challenges, the first thing that you have to do when you do this Luminex approach is you have to make sure that the drug itself has to be involved in the detection and capture steps. Otherwise, you run the risk of just measuring human IgG in your sample, which you don't want. So you have to devise a scheme where purified drug, in this case, it was mostly biotinylated Humira, was used in all of the assays. And you'll see that both in the capture steps, and you'll see that in the detection reagents that we use biotinylated Humira, both for the isotyping and the screening assays. That's to make sure that you're only getting drug-specific antibodies to generate your whatever signal in whichever assay it is. So that was something that we had to kind of rethink through. And that differs a little bit from the bridging assays that you see on the MSD and the colorimetric ELISA assays. And this data set shows that it does work. It works fairly well, and it has comparable sensitivity and drug tolerance to what you would get with these other types of technology. So it came out really nice. We're really proud of it. We're hoping that others would see what we've done, and to show you that you're not just limited to a colorimetric ELISA, and you're not limited to MSD. You can do these assays on other platforms, and there is a way to multiplex them and kind of conserve sample at the same time. So that was our main goal here. So
Great. Thank you for that, Paul. We have a few more coming in, and thanks again to our audience for submitting all these interesting questions. With this approach of screening and isotyping at the same time, has the regulatory body been approached as an acceptable method to approach immunogenicity?
yeah, so we have not used this and presented it to a regulatory agency. This was just sort of a proof of concept round. So there's sort of a good news, bad news with combining these two together. The good news is that you can do it and it does work, and you can get more information quickly. Sort of the bad news, and particularly when you deal with regulatory agencies, you may be generating more data than you're being asked for. So you run the risk of, well, we had a great screening assay, and now we showed that there's an IgM and an IgG4 response or an IgG1. You will probably be asked to do subsequent experiments to follow up on that you normally wouldn't have to do if you just did a screening assay. So this may not be the right approach for every single drug program, but if you know that there is going to be an interest from the regulatory agencies to look at specific isotypes, you can build that in these screening assays and generate that data early. So you really should have a conversation with these regulatory agencies on your assays before you use them, to make sure that it's the right assay and it answers the right question. So no, we have not approached them, but they love data and they love good scientific approaches. So if your assay is validated and developed well, I'm pretty sure that they're going to be really interested in it. But you still need to have that conversation if it's the right thing for your program.
Great, thank you. Looks like we have time for just one last question here. What do you foresee as the next steps in the development of multiplexed immunogenicity assays, and how could they further improve patient monitoring and drug safety?
Yeah. So I think what you have to consider is the industry is fixated on these single assays and this tiered approach, because that's the way it's done. That's what the regulatory agencies have seen, and that's the way everybody's been doing it. What we're trying to do is to open up your minds in that you don't have to do these in a sequential manner. You can multiplex these, and you can get data faster and more efficiently. Um, if there's a safety concern with some of these drug programs, or a particular safety concern with anti-drug antibodies for a given program, using a multiplex approach might get you there faster and get you there with better data, more, so you'll have more data to complete that picture for whatever questions being asked. But there are certain drug therapeutics out there that have a concern, and you know that you're going to be asked for isotyping data and other neutralization data. And sometimes using these, you can use a multiplexed approach, and if you can get to those questions quicker and more efficiently, um, that's always a better way of doing things. So it might not be the answer to everything, but it is certainly something that you have as an option. So think of these Luminex multiplex assays as part of your toolbox, right? You can do the traditional sequential single assays, but if you can also use a multiplexing approach. So it's kind of a mixed answer there, but I still think that the industry needs to consider that there may be other ways of doing this that may be more efficient, and this may be one of them.
Awesome. Well, that concludes our Q&A. So thank you again, Paul, for your time and for your important research. Any questions that we didn't have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you used to register. This webcast can be viewed on demand for 1 year until October 16th, 2025, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Thank you again to our audience for joining us today and for all of your interesting questions. And we encourage you to continue to explore this incredible virtual space. Until next time, goodbye, everyone. And Paul, thanks again.
All right, you're welcome. It was a pleasure. Take care.
All right, bye, everyone.